Hello everyone, welcome to the part 4 of Basics of Cancer, or Basics of Oncology from Nurture Oncology. And in the last video, we reviewed about cell invasion, certain steps in cell invasion, and the current research and future directions where you can go into. And a similar pattern today, we're going to discuss about metastasis. Metastasis is basically cancer spread beyond its original tumor location, and the secondary malignant growths in different areas at a distance from primary site of tumor. So either of the definition, you can go ahead with it. Here's a metastatic cascade diagram. This has been taken from Robbins, the Standard Book of Pathology. And the initial 4 steps were explained in cell invasion of how the cell differentiates, what loosening of the junctions and then invasion into the extracellular matrix. So today we are going to start with intravision, which is essentially the entry of the cell from its primary site into the blood vessel. So here you can see in the step marked as 1, tumor cells entering into the blood vessel and lymphatics. After entering into the lymphatics, there's certain interaction with the host lymphoid cells. Here, where you can study is into the tumor-associated macrophages, which we would talk upon later. After interaction with host lymphoid cells, so many of the tumor cells are destroyed here, undergo apoptosis. Some of them evade this mechanism by forming clumps with platelets. It's a tumor cell embolus, and this is how the tumor cells survive in the blood vessel, and they go from the primary primary tumor, to other organs. Then the tumor cell embolus goes and adheres to the basement membrane and to the basement membrane of the extravasation site in the organ there, which it has to extravase. And extravasation again is the exit of the cell from the blood lymphatic to the targeted area. So the first organ where the blood vessel or the tumor cells encounter through the blood or the blood spread is called the first pass organ concept. It is lungs in breast cancer and most of the thoracic cancers, as the lungs are where the major venous blood drains, and it is the primary area of metastasis, like in most of the cases, and the liver in gastrointestinal tumors. Now, the intravasation is comparatively easier than extravasation because the angiogenetic vessels which develop at the primary tumor area are leaky vessels which have space in between, which makes it easier for the cells to go into the lymphatics. Whereas in extravasation, the angiogenesis happens after the cells enter into the organ. So they have to extravase through the preformed, toughened vessels in the body, and therefore intravasation is comparatively easier than extravasation. Then after extravasation, there are metastatic deposits. These can be dormant or these can be actively dividing. And then angiogenesis, there are certain factors that promote angiogenesis. And during extravasation, there's interaction of the tumor cells via E-selectin, and this is a target of several trials. After angiogenesis and metastatic deposits, we have certain metastatic suppressors which are not being expressed. That leads to increased growth of the secondary tumors. Now, let's talk about the areas or the topics which under these steps which are being studied by researchers. So first, I would like to talk about CSF1 receptor blocker and macrophages on tumor-associated macrophages. So macrophages release certain cytokines which guide the tumor cells toward the secondary metastatic places. So CSF1 inhibitor, these have been approved by FDA recently in 2019 for tenosynovial giant cell tumor. So I was able to find out this study on tenosynovial giant cell tumors. There might be other type of cancers too, since this was approved in in 2019. So within a gap of 5 years, there have been studies on more solid tumors. Then another TAMs are also being studied as a biomarker for prognosis of breast cancer, ovarian cancer, and colorectal carcinoma. Then certain studies also claim that it can be used to monitor the real-time monitor of cancer and immunotherapy response via liquid biopsy. Liquid biopsy technologies are emerging in the field of research, cancer research diagnostics, and this would be a non-invasive process, as we would just see how much expression of TAMs is there, and if the immunotherapy is working, then TAM expression would be less and metastasis would be less. Then another is the circulating tumor cells, or when we talked about the clumping of the cells with platelets. So this is another topic, or this is the another step which have been under research and a potential area for targeted therapy. So as tumor cells form clusters with platelets, one trial and certain trials are there which are evaluating the role of platelet-derived platelet factor 4 for its metastatic potential. And there's also, people are also diving into the nanotechnology, and they are using liposomes and gold to break up the tumor cell clusters and epithelial to mesenchymal transition markers to prevent metastasis. So this was my area of interest, and this nanoparticle technology attracted me a lot. Then the third area is microfluidics and the use of digital PCRs and NGS to detect micrometastasis via these CTC clusters, and the quantification of these clusters to correlate with the aggressive nature of the cancer. Sorry. So this was CD45 and Epub, some technologies which I found out, and the PubMed and certain studies related to it, and these are being assessed for real-time assessment of metastasis. Here are the research questions which I formed. I thought that these would help you coming to your area of interest, and this is how you can form research questions based on the information I provided above. So what is the correlation between circulating tumor cell clusters and the progression of metastasis in breast cancer? And how do platelet levels correlate with the presence of CTC cluster in lung cancer patients? So this is how you can proceed, and then the density of CTC clusters in blood can predict metastasis risk in ovarian cancer. And what are the key biomarkers associated with the CTC clustering in a specific cancer type? So this is how I narrow down topics, or I brainstorm topics and derive on the possibilities in my institution, whether I can do a cross-sectional study or whether I would be allowed to do a cohort study or not. Then another step is the first pass organ concept, as I explained before. Then here researchers are again studying or focusing on the contribution of platelets in first pass organ metastasis in metastatic melanoma specifically, and they're evaluating whether the use of aspirin clopidogrel would be useful in metastatic melanoma or not. Then they one study, this was published in Nature 2023. I'm not sure, but most of the articles taken and which the topics which I'm giving you information about are taken from Lancet, Nature, Frontiers, and the top tiers in journals, so you can trust the source. I'll also give references of all the articles. Then this study um, this study was basically seeing the mechanical and biochemical factors which uh, help in the lung selective trapping of CTCs. What makes lungs more like help them to trap these particles and progress those tumor cells into tumorigenesis or metastasis. They use flow cytometry to assess cell-to-cell adhesion in RT-PCR here to see the DNA quantification, RNA quantification. Further, similar thing was applied in liver endothelial cells and Kupffer cells in colorectal carcinoma. The above study was done in breast cancer. Now, another attractive area, if you are, you can possibly find an oncologist as a guide in your institution, it would be great, and you might pursue this another area which I'm telling you. There was a study published to see whether immune checkpoint inhibitors, that is PDL1 inhibitor, whether they can modify the immune landscape of the first pass organs like lungs and liver and prevent early metastasis from the primary area, from the primary tumor. So, in a cross-sectional study in this aspect would be you can collect samples from the ongoing clinical trial or patients using PDL1 inhibitor or any immune therapy and see whether that correlates with the CTC level in the blood. This is a bit extensive study and is suggested for postgraduates. But if you are in an institution which promotes research actively, this is the area which I would suggest you to go forward because this is really interesting. And from an undergraduate's point of view, this would be an exceptional study and a new one. Then another areas are angiogenesis and growth of tumor. So there's a phase 2 trial going on to evaluate the potential of anti VEGF antibody as a therapy in non-small or non-small cell cancer, lung cancer, and melanoma. Then there's DCMRI study. This is a diagnostic study to see whether this non-invasive procedure uh, as an imaging biomarker for treatment in responsive breast cancer is useful or not. So they basically assess the angiogenesis in breast cancer using dynamic contrast enhanced MRI. Then we have phase 3 of another drug, aflibercept, combined with chemotherapy for platinum-resistant ovarian tumors. So why I'm telling you these trials are if you're a person who's into meta-analysis, or if you're a person into reviews, you can couple of these trials and publish a meta-analysis, start with the team and work towards systematic reviews on these topics. Then we have certain angiogenesis-related biomarkers. I don't think so this is too novel because VEGF and SVEGFR2 are being studied extensively. But if you could narrow it down and go towards some specific tumors, you might find a less amount of studies to sort of review on. Then metastatic growth is dependent on the metastasis suppressors. So if we restore the suppressors, that growth will decrease. And one of the gene which regulates that is NM223 gene, and it is and the agonist for progesterone and glucocorticoid receptors decreases the NM23 gene. That is helpful in restoring metastasis suppressor. This was also seen in a study. It's in phase 2 trial in metastatic breast cancer. You can see the effect of these agonists for progesterone receptors and glucocorticoid receptors and other cancers as well. Narrow it down to your area of research, whether such a topic exists or not. This breast cancer definitely exists. That's why I've written it here. So this was all about today. I hope you got a lot of topics to think upon, to wander on, and let us know if you have any doubts. Thank you so much, everyone. Have a good day.